postdoc at University of Texas, Austin. Before we begin, I just wanted to warn you guys, this is going to be an R-rated presentation. I don't expect any violence or grisly images, but there could be some sexuality and definitely foul language and maybe even some nudity to talk to as well, so keep that in mind. So one major discovery we've made recently in microbiome research is how variable the microbiome is, both among populations, among individuals within populations, and even within individuals through their lifetime. And we're now searching for the factors that are responsible for generating that variation. And I want to talk about two of those factors with you today. The first are social interactions. There's been a number of recent studies that have shown that who individuals interact with and how often they interact with those individuals has an influence on their microbiome. So social interactions is emerging as one of the factors that might influence this variation that we see. The second is diet. There's been some really fascinating work done looking at how diet affects the microbiome. For example, this study by Lay et al. Looked at the feeding habits of various mammals and found convergence in the microbiome amongst, depending upon whether they're eating herbivores, whether they're carnivores, whether they're omnivores. And this other study on the right here by David manipulated diet in humans, either with a plant-based diet or an animal-based diet, and found that particularly in this animal-based diet, which is a high-protein diet, he found rapid changes in the gut microbiome by performing this experimental manipulation of what people were eating. And in terms of social interactions, a really understudied social interaction is mating. Despite its ubiquity, most studies looking at the microbiome with respect to mating have been focused on sexually transmitted infections. But what we know now is that many sexually transmitted infections are actually beneficial. If you're interested in that, you can see this paper in Tree that was published last year. And in addition, social interactions, including sexual interactions, also have effects on host physiology. And so that can influence host microbiome interactions. Finally, sexual interactions also can involve diet. Before you think I'm going to talk about any kind of freaky, perverse, kinky sex here, what I'm actually talking about is particular cases where males feed their mates. So nuptial gifts are any non-gametic material provided by a donor, usually a male, to a recipient during courtship or copulation. And they come in many different forms. Sometimes males provide prey items they capture for females. It could also involve cases where males transfer substances to females in seminal fluid. It can involve situations where females eat the whole male, like in spiders. Or the particular case I'm going to focus on today are glandular gifts, where males secrete something that females then consume after copulation occurs. So glandular gifts are these proteinaceous secretions that males produce. They are very variable in size among species, which is a question in its own right. So this is a frequency histogram showing spermatophilic size and the number of species. And they also have variable effects on female fitness. This has been a long-standing question in evolutionary biology of why this is the case. It's really the topic for another talk. But for the purposes of this talk, there's really a couple things that I want you to know. So first of all, the spermatophore, particularly in crickets and katydids, is composed of two components. There's the ampulla here, which is the sperm-containing part, and then this is the spermatophylax, which is the part that the female actually consumes after the female mounts the male, and then he takes off and she eats this nutritious meal. So they vary between species and size. So here's the spermatophylax of this guy here. So this is the female eating this after mating, and this is another female here, and you can see the spermatophylax is very diminutive in this particular species. So I thought this would be an interesting contrast to try and look at how sexual interactions affect the microbiome, using this variation in nuptial gift size, and also thinking about how mating itself might affect the microbiome independent of the consumption of these gifts via, for example, sexual transmission or through changes in host physiology. So the two species I looked at to perform this study were the decorated cricket, Rhodotus sigillatus. This species has very small nuptial gifts. They're about 5% of the male's body weight, and they're really just large enough for the sperm to complete their transfer. They have no effects on female fitness at all. So our current impression of these particular gifts is that they're really just large enough for the sperm to be transferred, and they don't serve any role in terms of providing females with nutrition. So it's kind of like a sensory trap for females. The second species that I chose to look at was the Mormon cricket, an average simplex. These katydids actually are known for the production of large gifts, about 19% of the male's body weight. You guys can imagine having to do that every time you mate. It takes these guys like 3 or 4 days to recover from that. They are known for two things, really. These biblical outbreaks of marching crickets that form these marching bands across the western United States. It's driven by their hunt for protein and salt, because they become nutrient-loaded in those environments. And also, there are well-known cases of nutrient-dependent sex role reversal. And so every behavioral ecology textbook has this example of Mormon crickets. When food is very plentiful, they have these traditional sex roles. Males compete for females, and females are the choosy sex. Well, when things get bad and the nutrients are scarce in the environment, it's the females that are competing for these males that produce these spermatophylaces because they're starved for protein. And so females become the competitive sex, and males become the choosy sex. And for these reasons, we believe that Mormon crickets are producing a nuptial gift that's beneficial to females and provides lots of nutrients. And that forms the basis for the study that I want to tell you about today. So the two main hypotheses I was interested in testing are, first of all, the spermatophylax affects the composition of the microbiome. So the reason why this might be the case is because the increase in dietary protein alters nutrient input into the gut, and therefore alters the substrate on which microbes in the gut subsist. And this is one of the main mechanisms that we think that diet influences gut microbes. The second is that mating independent of spermatophylax consumption could also affect the microbiome. And there's a couple ways that could occur. One is through sexual transmission, and the second is through physiological responses to mating affecting the host microbe interactions. And that's kind of the framework from which I've been working on my postdoc. So in terms of the study design, we collected 7th instar katydids and crickets from the field. We allowed them to undergo their adult molt in the lab, let them mature to have virgin females and males after 6 to 10 days, and then we took a fecal sample. Following that, we exposed them to one of 4 treatments in a 2x2 factorial design. So that's shown here. So females were allowed to either mate or not, or consume spermatophylax or not. They underwent that treatment for 12 to 14 days, and then at the end of the experiment, I took another fecal sample, and I also dissected the spermatheca, the ampulla, and the spermatophylax, and subjected all those to DNA extraction, 16S Illumina sequencing, and then looked for OTUs at 97% similarity, and used green genes to get the taxon ID. For the analysis, I used a constrained principal components analysis, and so for the Mormon crickets, I calculated Bray-Curtis distance, looked at the effect of mating spermatophylax consumption, and also time. Remember, there's 2 time points here, a pre and a post experiment. With the decorated crickets, it was the same exact thing, except for in this particular species, I also had a diet treatment where I supplemented females that didn't mate or consume the spermatophylax with protein to look at the effects of protein consumption independent of these social interactions. So what did I find? Just to orient you here, I've got the data set up into columns and rows. So this first row here is the pre-experiment samples, and the second row here is the post-experiment samples. The left column is the spermatophylax consumption, and the right is mating or not mating. And so each of these points is an individual female, and they're colored by what treatment they were assigned to. And so what you can see in this principal components plot is that before the experiment began, as you might expect, there was no difference between treatments in the composition of the microbiome. After the experiment in the decorated crickets, as I expected, there was no difference amongst females that consumed spermatophylax or did not consume spermatophylax. And this was expected because, as I said earlier, they produce very small gifts, and so I didn't necessarily expect to see a strong effect here. Now, with respect to mating, I really didn't know what to expect, because we know very little about what males transfer, or in addition, what effect mating has on host physiology in either of these species. So I wasn't really sure what to expect. In this particular case, actually I didn't find any effect at all either. Thirdly, I didn't find effect of diet. That's not shown here. So in this particular species, no effect of spermatophylax consumption, no effects of mating independent of spermatophylax consumption, and no effect of protein. So it seems that that it's possible that the microbiome of these guys might not be responsive to protein at all. As I indicated earlier, it that could be a reason why there's no effect of spermatophytes consumption, or it could be they just produce really wimpy gifts. So disentangling those two would require another experiment. What about the Mormon crickets? Sorry. So surprisingly, I did not find this effect of nuptial gift consumption in Mormon crickets either. And in this particular species, we didn't have the logistical capacity to do the protein supplementation experiment, so I'm not sure if we didn't find an effect because the nuptial gifts are wimpier than they're expected to be given the evidence that we have, or if it's because their gut microbiome as well isn't responding to the protein input, as has been found in mammals. So that's an avenue that I'm interested in pursuing to find out which of those is the case. Interestingly, however, I did find an effect of mating independent of spermatophytes consumption on the gut microbiome. And so the next question then becomes, which of those OTUs are the ones that are responding to the treatment? So we'll talk about that first. So this is a ordination of the OTU scores on that same plot I just showed you. And what you can see here is that along this x-axis, which is associated with either mating or not mating, so each of these individual points are the female scores, and then the triangles here are the individual OTUs. We find that there's a cluster here of about 5 OTUs that appear to be responding to whether a female mated or didn't mate. And a couple things to note about these. First of all these OTUs are lactic acid bacteria from the genus Pediococcus or Lactobacillus, and particularly these top two Pediococcus occur at high abundance in the samples that we have. This one in particular is the third most common OTU that we find in the gut. So these aren't diminutive OTUs that are occurring at low abundance. It suggests that changes in the abundance of these OTUs might actually have important fitness consequences. Lactic acid bacteria in general are these facultative anaerobes that are associated with mucosal surfaces in lots of animals, in addition to other environmental places. Overwhelmingly, the literature suggests they have beneficial effects on their host. They're used as probiotics, they accelerate development in flies through nutrient signaling, and they provide protection from pathogens in a number of different ways. And so what my data suggests then is that mating is increasing the abundance of these beneficial microbes. And so while that is the focus of my continuing work as a postdoc to evaluate that, at least suggests that mating is promoting these beneficial microbes in the gut, which is really interesting, and the first time this has ever been shown to my knowledge. Why might mating affect gut microbes? That's kind of the second question, and I've kind of outlined this already previously, but to go into a little more depth, I think it's interesting to think about why social interactions in general, not just sexual interactions, affects the microbiome. And of course, the traditional route thought of is related to transmission. And this can occur through a variety of points in time in the mating interaction between males and females. But I think understudied or underappreciated route might be through host physiology. And the reason I think this might be the case in my particular system is all the reproductive tissues that I looked at were for the most part sterile. And so I think sexual transmission is actually unlikely in this particular system. I think changes in host physiology might be the key to understanding why I observed the change that I did, and that's my current focus in my own research. So with that, I'll take any